In this video, we're going to talk about gene expression, which is how a living organism is built from the genetic code. This is the fourth video in a series on DNA. So remember that DNA is a blueprint for both physical and behavioral features, and genes are the basic unit of inheritance that transmits information from parents to offspring. Now, genes code primarily for proteins, and proteins control the building and all the other activities within a cell or within an organism. The analogy I like to use is proteins are the little robots that get everything done inside the cell. Now, the rule of thumb is that one gene codes for one protein. So a gene is a very small section of DNA that codes for one protein. It's not actually as simple as that, but it's a good rule of thumb to start with. So DNA specifies your physical traits, your features, by dictating protein synthesis. Proteins are the links between your genotype, or what's in your DNA, and your phenotype, or your physical expression. So gene expression is the manifestation of the message that's contained in the genetic code. The molecular chain of command is from DNA in the nucleus to RNA, which exits the nucleus and is just floating around in the cell's cytoplasm, and then from RNA to protein. So that describes the central dogma of biology. The central dogma of biology is that flow chart. The genetic message begins as DNA, gets transferred to RNA, and then gets expressed as protein. So there are two steps in that central dogma. The first is transcription. Transcription is the synthesis of RNA under the direction of DNA. And then the second step is translation, which is the synthesis of proteins under the direction of RNA. The easiest ways to remember these names is to think about what these words mean colloquially. So what does it mean when you transcribe something? It means that you're copying verbatim. You're copying word for word. If I were transcribing the court proceedings for a court case, I would be copying what was said word for word into a court transcript. So remember that DNA and RNA are very, very similar molecules. So you can copy a DNA code verbatim into an RNA code. It's the same language. But protein and nucleic acids are completely different molecular languages. RNA and DNA are both nucleic acids, but protein is built out of strings of amino acids. So those are entirely different molecules. So this is a different language. And when you convert a message from one language to another language, you call it translation. You can translate a message from Spanish to English. I just want to quickly remind you about all the important functions of proteins. And this is review from previous lectures. They're important for structural support. For example, the cytoskeleton inside the cell, the scaffolding that holds the cell's shape, is made out of proteins, but also the linkages between cells. They are also made out of protein. Communication. So cell-to-cell communication is often performed with proteins. Neurotransmitters can be proteins, hormones can be proteins. The receptors that receive the chemical signals are also proteins. So proteins are very important in allowing the cells from different parts of your body to communicate with each other and coordinate their activities. Defense. Antibodies are made out of proteins, but they're not the only defense molecules. Your cells make a variety of endogenous antibiotics. Many of those are small peptides or little proteins. Portals across the cell membrane. The cell needs to bring in nutrients, it needs to excrete waste, and it transports those things back and forth across the cell membrane through protein channels. Movement. Your muscle fibers are made out of protein. Flagella on sperm or on bacteria, they are also made out of protein. So proteins are essential for cells to be able to move around as well. And then, of course, very importantly, enzymes. Enzymes catalyze chemical reactions inside the cell. They change molecules from one molecule type to another. Chemical reactions are important for building large molecules out of smaller ones, or for deconstructing larger molecules into their smaller parts. The monomers of proteins are amino acids. Here's an example of one. And there are 4 levels of protein structure. The primary structure of a protein describes the sequence of amino acids, just as if they were strung out in a straight line. Remember, there are 20 different amino acids. So an example of the primary structure of a protein would just be, for example, methionine, leucine, tyrosine, serine, and so on and so forth. So it's just the sequence of the amino acids as they're strung together in the polypeptide. The secondary structure is formed by hydrogen bonds between carboxyl groups and amino groups of non-adjacent amino acids. So, for example, one of these amino acid, this amino acid here, this carboxyl group here is forming a hydrogen bond with the amino group of this amino acid down here. And these hydrogen bonds that hold together the secondary structure of a protein can form shapes like alpha helices, which are what are shown here, or beta pleated sheets. Now, the tertiary structure of a protein describes this complex three-dimensional folding pattern that's held together by interactions between the R groups of the different amino acids. So remember, each amino acid has an amino group, a carboxyl group, and a mystery R group. The R group is what differs between the 20 different amino acids. So each of these amino acids in this chain will have a different R group, and each R group has its own chemical properties. Some are positively charged, some are negatively charged, some are polar, some are nonpolar or hydrophobic. And so the sequence of amino acids will dictate how these R groups interact with each other and what shape they form when they fold up upon themselves. So if you were to change out one amino acid for another in the primary structure, it could change which amino acids are interacting with which in the chain. And that could change the three-dimensional shape or the tertiary structure. Quaternary structure is a lot like tertiary structure. It's held together by R group interactions between different amino acids. The difference is it's between separate polypeptide chains. So the molecule shown here is hemoglobin. Hemoglobin has 4 subunits, 1, 2, 3, 4. Each one is a separate chain. Each one has a tertiary structure and has folded up upon itself, but now they're also attached to each other through these R group interactions. Now, the sequence of nucleotides in DNA provides a code for constructing a protein. Protein construction requires a conversion of a nucleotide sequence to an amino acid sequence. Now, in transcription, we rewrite the DNA code into RNA using the same nucleotide language. It's transcription because the message is copied verbatim, and translation involves switching from the nucleotide language to the amino acid language. So here's a diagram of the overall process. We will go into detail about each individual step, but you can see here's the DNA code here. Remember, DNA is huge. It's the whole genetic library, and it's double-stranded. RNA is short, and it's single-stranded. So this is a little transcript or photocopy of a small segment of DNA, of one gene of the genetic library. And then here is the translation part where the nucleic acid code is translated to an amino acid code. You can already see that in RNA, the genetic message is divided into 3-letter words. Okay, you can think of this genetic message as built out of 3-letter words. Every 3 letters of the genetic code codes for one amino acid in the protein. All right, so let's look at transcription in more detail. Transcription is very similar in many ways to DNA synthesis. The DNA has to be unzipped. A complementary strand of RNA, this time, is built using the complementary base pairing rules. It's also built in an anti-parallel fashion. So there are many similarities between DNA synthesis and transcription. So let's take a look. I'm going to draw the parent DNA in red, and I'm just going to make up a genetic code for a tiny little gene here. I'm just making something up. This is not a real gene or anything. And of course, there's 2 strands of DNA, and they're complementary to each other. So A's match with T's, and G's match with C's. Okay, now the first step of transcription is that RNA polymerase comes and unzips the DNA and builds a complement to the template strand. And it builds it using a DNA transcript. So it's going to read one of the parent strands and build a complementary RNA transcript. So let's just say that this is the template strand on the top. So let's separate these 2 strands, and now let's build the RNA transcript. So the template strand, I said, is the top strand. RNA polymerase reads the DNA 3' to 5', and it builds an anti-parallel transcript in the 5' to 3' direction. So 5' in the RNA matches with a 3' end in the DNA. And we are going to follow the same base pairing rules. T's bind to A's, but this time, remember I said that RNA doesn't have thymine. Thymine is replaced by uracil. So everywhere you would have put a T in a DNA complement, you have to replace it with a U in the RNA complement. So there we go. But the rest of the rules are the same. C's to G's, G's to C's, G to C, G to C, A to U this time. But T's in DNA still bind with A's in RNA. A to U, G to C to G, C to G, C to G, 3'. Okay. Almost exactly the same as DNA synthesis. If you look at the RNA transcript and the non-template strand of the DNA, you'll notice that they are exactly the same, except all the T's in DNA have been replaced by U's in RNA. All right, so let's add to our notes. Travels 3' to 5', builds 5' to 3'. Okay, so how does RNA polymerase know which of the DNA strands is the template strand and which is the non-template strand? It's because every gene is preceded by a DNA sequence called a promoter. A promoter is a sequence of nucleic acids that tells RNA polymerase bind here. I'll never make you memorize the promoter sequence. They're much too long for that. You just need to know that every gene is preceded by one. So if we were to look at a long strand of DNA, so let's say this is a parent strand of DNA, and let's just say the gene is here. This segment is a gene, code for a protein. That means that right here is the promoter sequence. This tells RNA polymerase to bind here. And every gene ends with a sequence called a Terminator sequence. The Terminator sequence tells RNA polymerase that it's done, and RNA polymerase releases once it reaches the Terminator sequence. So RNA polymerase is going to travel along one of these strands in the 3' to 5' direction. And once it reaches the Terminator sequence, it will release. So the start of the gene is marked by the promoter sequence and the end of the gene is marked by the Terminator sequence. So between the promoter and the Terminator is where the RNA transcript is built. And remember, the RNA transcript is built through complementary base pairing rules, and it is anti-parallel to the DNA template strand. And that's transcription. RNA polymerase binds to a promoter sequence at the start of a gene. RNA polymerase unzips the DNA and creates a complementary anti-parallel RNA strand from the template DNA strand. Complementary base pairing rules apply, except that T is replaced by U in RNA. RNA polymerase travels the template DNA strand from 3' to 5'. The RNA transcript it builds is anti-parallel. It's built 5' to 3'. Once RNA polymerase reaches the Terminator sequence, it releases. Here's a prettier diagram of this happening. So in this image, here's RNA polymerase, and it binds to a sequence of DNA called the promoter, and it unzips the DNA as it travels. The DNA rewinds itself behind RNA polymerase as RNA polymerase moves down the gene. This strand down here is the template, and it's separating the template from the non-template strand, and it's adding RNA nucleotides as it goes, and once it reaches the Terminator sequence, it detaches. Okay. And now, in pink this time, you have the completed RNA transcript. So this is an up close of what is included in that RNA transcript once that transcript leaves, once it's complete. So this whole thing is the RNA transcript. Here you have the start of the genetic message. This is destined to be translated to protein. But before and after the genetic message, there's still some RNA. So what is that? There's the cap and the tail in particular. The cap and tail are junk sequences. It's just junk. So why are they there? They're mostly there to protect the genetic message from damage. So like if you have a shoelace or a piece of thread or a rope, you might notice that the ends can get frayed while the middle stays in better condition. So there's junk DNA that's not needed on the ends, and that's just there as a buffer. You don't want the actual genetic code that's being carried by the RNA to be damaged. So this whole piece here is junk, protection from damage. It's the same with this segment here, except the cap also helps feed this RNA transcript into the ribosome, which is the machine that builds the protein from it. So there has to be a little extra RNA at the front end to help feed the RNA into the machine that's going to build protein from this genetic message. You can click on the video link to see a video of transcription happening in real time. Now, there are 3 types of RNA transcripts, and while all of them are involved in building protein, not all of them are actually a blueprint themselves for protein. So let's take a look at the different types of RNA transcripts that are made through transcription. The first kind is messenger RNA. Messenger RNA is a photocopy of a gene in the DNA, and it is a blueprint for building a protein. Now, rRNA stands for ribosomal RNA. Ribosomal RNA actually builds the ribosome, which is the protein construction machine. Now, ribosomes are enzymes. They catalyze chemical reactions. They string amino acids together into long chains. Now, most enzymes are made out of protein, but ribosomes are a little bit different. They're actually made out of a mix of RNA, ribosomal RNA, and protein. So the ribosome reads the messenger RNA and uses the instructions in the messenger RNA to build a protein. tRNA stands for transfer RNA. Transfer RNAs float about and they carry amino acids, and they bring the amino acids to the ribosome in the correct order so that the primary structure of the protein can be correct. Here's a cartoon image of a transfer RNA. So this is more cartoony, this one on the right here. So at this purple part is where the amino acid would be. So the amino acid is bound to this transfer RNA. And at the bottom, you'll see there's something called an anticodon. The anticodon is just a series of letters of nucleotides, and they bind in a complementary fashion to the strip of messenger RNA that's in the ribosome at a particular moment. And here's another image that's a little less cartoony. And in this image on the left, you can see here's where the amino acid would bind, the amino acid attachment site. And then this is a long strand of RNA, and like all RNA, it's single-stranded, but it's folded up upon itself, kind of like a protein folds upon itself. And its structure, its shape, is held together by hydrogen bonding, complementary base pairing between different nucleotides within this one chain. We call that internal complementary base pairing. And still you see the anticodon here. The anticodon is really important for the transfer RNAs to be able to bring the amino acids in the correct order. So here's a general overview of translation. The flow of information from gene to protein is based on a triplet code, meaning the genetic instructions for the amino acid sequence of a polypeptide chain or a protein chain are written in both DNA and RNA as a series of non-overlapping 3-base words called codons. So for example, a codon could be in RNA AUG, or it could be UUU. These 3-letter words are what indicate which amino acid comes next, and they are called codons. Each amino acid is specified by one codon. Now, there are 64 possible codons, because what we're thinking of, so how many letters are there? There's 4 letters, right? The 4 letters are, in RNA at least, A, U, C, G. And then we have 3-letter words. So how many ways can you fill 3-letter words using these 4 letters? The answer is 64. There's 64 possible codons, but we only have 20 amino acids, which means that we have some degeneracy in the genetic code. There are several codons that all code for the same amino acid, so there's repetition. And I'll show you what I mean. So this is a codon chart, and it decodes the messenger RNA, the mRNA. So if you have a strand of messenger RNA, you can look at a codon chart and figure out what protein it codes for. So for example, let's just say that your message started AUG. So you would look through this codon chart for the AUG. So you'll notice the first bases are listed in this column here. So my first base is A, so A is here, right? There's A. So I would want to look in this row for my codon. And then the second bases are listed as the column headings. So U is my second base. So here's U right there. So I'm looking at this column. So the third row and the first column, and I want to know where they intersect. They intersect here, right? And then the third is G. So in here, I just look for the AUG, and there it is, methionine or start codon. So in eukaryotic cells, all proteins start with methionine. AUG is the start codon. It tells the ribosome where to start. Just like the promoter told RNA polymerase where to start, the start codon tells the ribosome where to start. So AUG codes for, it indicates the start of a protein, and it also codes for methionine. Let's just say my next codon in my messenger RNA was GGG. Okay, let's see what that codes for. So let's erase what I did so far, and let's start again. So again, the first base is the row headings. So G we're looking for. So here's the row of G. And then my second base is the column headings. So I'm looking for G again. So the fourth row and the fourth column, and I want to look at where they intersect. They intersect right here. And GGG is right here. That stands for glycine. Now, here's where you see the degeneracy in the code, because GG anything codes for glycine. GGU, GGC, GGA, GGG, they all code for glycine. And that, again, is because there are 64 possible codons, but there's only 20 amino acids. So multiple codons can code for the same amino acid. Let's look at UAU. U is the first base in this codon. There it is. So we're looking at the top row here. The second base is A. That's here. You know, the second base is going to be represented by the column headings. So this is the third column. And we'd want to see where the first row and the third column intersect. That's here. UAU is tyrosine. So let's just say the next codon is UAA. So again, U, first row, A, third column. This time it's UAA. That's right here. And that's a stop codon. That tells the ribosome to release. So the start codon tells the ribosome where to begin translation, and the stop codon tells the ribosome where to end translation. Running out of space over there. So the start codon is to the ribosome as the promoter is to RNA polymerase. The stop codon is to the ribosome as the terminator is to RNA polymerase. So now let's look at some diagrams of translation. Here's the ribosome. The ribosome has 2 subunits. It looks like a hamburger, kind of. And the messenger RNA is going to fit between the large and the small subunits. There are 2 empty slots for transfer RNAs here. One transfer RNA will bind if its anticodon is complementary to the messenger RNA codon. And the same is true for the transfer RNA that fits in the second slot. Now, once 2 transfer RNA molecules are bound, remember they're carrying amino acids. And the ribosome is going to catalyze a dehydration reaction to link these 2 amino acids together. Once the 2 amino acids are linked, the first transfer RNA will leave, and the second transfer RNA will move over into the first slot, which means the first slot will be empty and another transfer molecule will have to arrive and will only bind if its anticodon matches with the messenger RNA codon that is now in that slot. So this ribosome is moving down this messenger RNA, pulling the messenger RNA through like a tape, and is reading the three-letter words, the codons that are in those empty slots as it moves down the transcript. So again, once two transfer RNAs are in the ribosome catalyzes a dehydration reaction linking the two amino acids that they carry. Then the first transfer RNA will depart, and the ribosome will move down the strand of messenger RNA to the next codon, and the second transfer RNA will move into the first slot. And a new transfer RNA will arrive in the second slot. All right, so we'll look at more images of this happening. So in this image, you can see the codon in the pink messenger RNA and the binding between it and the anticodon in the transfer RNA. Note how the complementary base pairing rules apply. A to U's, U's to A's, C's to G's. The ribosome comes together around the messenger RNA and the first transfer RNA. And here you can see how the ribosome is going to move down the messenger RNA. So let's start with number 1, where the second transfer RNA arrives. The ribosome catalyzes a dehydration reaction between the amino acids that these two transfer RNAs are carrying. The first transfer RNA molecule leaves, and it leaves behind the amino acid it brought to the chain. Now, this second transfer RNA has to move over into the empty slot. Now, the second slot is empty right here. And so a new transfer molecule can arrive, and the cycle continues. Now remember, all three types of RNAs that we discussed are here. So this is the messenger RNA in pink. It's carrying a photocopy of the protein code. The ribosome is built out of ribosomal RNA and protein, and these green molecules that are carrying the amino acids are called transfer RNAs. So again, all three types of RNA are involved in producing proteins, but not all of them are actually themselves the instructions for the protein. All right, so how do we keep, start codons, stop codons, caps, promoters, terminators, how do we keep that all straight? I'm gonna put it all together so you can see how it all fits together in one sketch. And I'm also gonna mention a few new pesky details that I failed to mention earlier. All right, so let's start with some DNA. Double-stranded, and we're just looking at a small segment of the DNA. We're looking at one gene, and every gene is preceded by a promoter sequence. We don't need to know what the promoter sequence is. If you were doing a lot of DNA sequencing, it might be relevant to you, but it's a longer sequence. We don't need to memorize it. Now, the promoter tells RNA polymerase where to begin, but remember, it doesn't start transcribing the gene right away because there's some junk between the promoter and the actual gene. That junk is the cap. And I'm just making up a sequence for the cap, just some random selection of letters. And of course it has a complement. The promoter also has a complement as well. And then we have the actual gene. And I'm just going to make up a gene. And the complementary strand has a complement to the gene. At the end of the gene is a cap, which again is just going to be some junk. I'm again just going to make up some letters for this. And after the junk is the Terminator sequence. So this is the DNA strand. The promoter is on the top. That means the top is the template strand. It's going to be running 3' to 5'. The complementary strand runs in the opposite direction. All right, so RNA polymerase, I'm going to make in light blue. RNA polymerase is going to bind to the promoter, and it's going to transcribe the gene, all of it, including the caps, because RNA polymerase is not designed to recognize the caps. It's designed to bind to the promoter and release at the terminator and transcribe everything that's in between those two. So let's now draw the RNA transcript that results. The RNA transcript is anti-parallel to the template strand, and the RNA transcript is complementary to the template strand. Remember, these first few nucleic acids represent the cap, and then we have the gene. And at the end of the gene, we have the second cap. So RNA polymerase ran along the template strand 3' to 5'. It started at the promoter and released once it got to the terminator. The transcript includes caps, which are just junk, and sandwiched between the caps are the gene. Now, this is going to go to the ribosome. And the thing I didn't mention before is that unlike RNA polymerase and DNA polymerase, the ribosome reads in the 5' to 3' direction. So RNA polymerase reads 3' to 5'. DNA polymerase reads 3' to 5'. Ribosomes read 5' to 3'. So this is going to be the start end for the ribosome. Here are both the large and small subunits. So the front cap at the 5' end is going to be used to feed the messenger RNA into the ribosome, and it's going to hang out the other side. So the ribosome will keep pulling this messenger RNA tape through until it finds the start codon. And if you remember from the codon charts, the start codon is AUG. So once the AUG gets into the ribosome begins translation. Now we're going to also have the second codon in the ribosome. The second codon here was GGG. So now transfer RNA comes to the ribosome and binds if the transfer molecule has an anticodon that matches with the codon through complementary base pairing. So the transfer RNA I'll do in green. The only transfer RNA that can bind there is one with an anticodon, that's UAC, 3', 5'. Always antiparallel, always complementary base pairing. And this transfer RNA always carries methionine. The next transfer RNA that can bind in the second slot has to have the complementary code CCC. And this transfer RNA always carries glycine. Once these two transfer RNAs are in the ribosome, they are bound together through a dehydration reaction, and the first transfer molecule leaves, the ribosome moves down, the second transfer RNA moves into the first slot, and a new transfer RNA comes down for the third codon position. So the third codon here was UCC, the fourth codon was UAA, and then we had some junk here, the last cap. So remember, these caps are ignored. I explained how the ribosome knows to ignore the first cap. It's because it hadn't reached the start codon yet. The reason it ignores the last cap is because the stop codon is right before it. So let's decode these. This third codon here, UCC, codes for serine. So serine will be added next. This last codon here is a stop codon. Once the ribosome reaches the stop codon, it releases, everything falls apart, and the whole process ends. So this is just an example. It's a very short protein, but it carries the primary structure of this protein is methionine, glycine, serine. That's the primary structure. It's just the sequence of amino acids. So this shows you that there's a huge amount of DNA. RNA polymerase knows where to find the start of the RNA transcript because it looks for the promoter sequence. It binds to the promoter and then runs along the DNA in the 3' to 5' direction and transcribes everything that's there. And then when it reaches the terminator sequence, it releases. What's there included junk, gene, junk. The first cap, the first segment of junk, is used to feed the messenger RNA into the ribosome. The start codon tells the ribosome when the junk or the cap has ended. The ribosome begins translation at the start codon, and it keeps translating until it reaches the stop codon. Then it releases, and the second cap or the second segment of junk is ignored that comes after the stop codon. So that's everything put together. If you'd like to see translation in action, I encourage you to click on this link. Now that we've talked about how DNA is expressed, we can talk a little bit more about mutations and exactly how mutations affect an organism. Mutations can be divided into two general categories. There are base substitutions and there are frameshift mutations. A base substitution is where one letter, one nucleotide, is traded out for a different one. So if you had the code AATAA and the cell was copying its DNA to its daughter cells and it read the message wrong and copied it wrong, it might be ATTAA. All right, this A just got traded out for a T. That's a base substitution. Now, a frameshift mutation is where a nucleotide is inserted or deleted. So now you have too many or too few nucleotides in the gene. So an example of that would be, again, if the original code was AATAA, and we added an extra one, AATAA. We just added an extra A into there, so now there's more nucleotides than there should be. All right, now focusing on base substitutions. Base substitutions can be silent, meaning that there is no change to the protein's primary structure, and therefore no change to the three-dimensional shape of the protein. To understand how this can happen, we have to go back to the codon chart. So if you had a DNA code that was 3' A, A, T, 5', that creates an RNA code that is 5' UUA 3', and UUA 3' codes for leucine. All right, so let's look at a base substitution where AAT is changed into AAC. So we've changed the T to a C. That's the base substitution mutation that this cell has experienced. This is going to change the RNA codon to UUG, and UUG is still leucine. So nothing has happened to the protein. This is what is known as a silent mutation. The genetic code was changed, but the protein wasn't changed at all. Now, the second type of mutation on our list was a missense mutation. A missense mutation is a base substitution that causes one amino acid to be changed to another amino acid. So I'll show you what I mean. Now let's say that AAT experienced a base substitution mutation in which the T was changed to an A this time. So now we have AAA. This is still just a change in one nucleotide, but this time it has a different result. The messenger RNA code is now UUU, and if you look at the codon chart, UUU now codes for phenylalanine. So wherever leucine was in this protein, it's now replaced by phenylalanine. And it's hard to predict what that would do to the resulting protein. Some of that depends on exactly where the mutation happened in the protein, and some of it depends on the chemical characteristics of leucine and phenylalanine. If you had a positively charged ionic amino acid that was replaced with a negatively charged ionic amino acid, well, those two amino acids have entirely different chemical properties, and it could really dramatically change the shape of that protein and therefore change its function. The last type of base substitution mutation to look at is called a nonsense mutation. In nonsense mutations, the original amino acid is changed to a stop codon, and that means that the protein is now truncated. So instead of the ribosome continuing on to add more amino acids to the growing polypeptide chain, it releases and everything stops and the protein is now shorter. So let's look at an example of how that can happen. So again, our original code was AAT in the DNA in red, but now the base substitution is going to happen in the second position of this codon. So this time the A in the middle position is switched out for a T. in this particular mutation. So what's the result in the RNA transcript? Now we have UAA. And I see I've mixed up my primes here. So what does this do to the protein code in translation? Well, what is UAA? UAA is a stop codon. So now you don't get leucine at all. You get a stop. And the protein's truncated. All right, so we covered our missense mutations where one amino acid is replaced with another, and we covered our nonsense mutations where an amino acid is replaced with a stop codon. So now let's talk a little bit more about frameshift mutations, insertions and deletions. If we have a genetic code, these are our codons in the DNA. Let's add one more here. Now in blue, I'm going to put our mutation. Let's start with an insertion. Let's say we add an extra A here. So that's an insertion. This is called a frameshift mutation because you can think of the frames like these lines I drew that separate the different codons. Well, they're all gonna shift over one because I've added an extra letter, an extra nucleotide. So originally we had GAG, now we have AGA. That's gonna be two different amino acids. Then in the original version, we had ATT, now we have GAT. Again, that's going to be an entirely different amino acid. Then we had in the original version ATA, but now it's TAT, again, a different amino acid. So everything after an insertion or a deletion is going to be changed. Deletions do the same thing, they just frame shift in the other direction. So if you have an insertion, everything shifts one way. If you have a deletion, everything shifts the other way. So it creates these cascading effects throughout the entire protein. Here's some practical real-life examples. So there is a disease called sickle cell anemia, and it's caused by inheritance of a mutant allele, a mutant version of a gene for hemoglobin. Hemoglobin is a protein that makes your blood red. Your blood cells are filled with hemoglobin. So here's a red blood cell. RBC stands for red blood cell. And a normal red blood cell has sort of like this donut-looking shape. It's depressed in the middle. And it's filled with this protein called hemoglobin. The hemoglobin grabs onto oxygen and carries it throughout your body. It also grabs onto CO2 and carries it back to your lungs so that you can exhale the CO2. So hemoglobin's really important for oxygen and carbon dioxide exchange between your body and the environment. A mutation to hemoglobin, there is a genetic mutation that some people have where the hemoglobin is an abnormal shape. And it causes the blood cells to take on a sickle-like shape. So this is an example of a red blood cell's shape when the hemoglobin is mutated with the sickle cell mutation. And it can create all kinds of problems in the body. It's called anemia. Anemia is any disease that prevents you from oxygenating all your tissues. In this case, sickle cell anemia is when the red blood cells have trouble traveling through your blood vessels because of their shape. You can see here, they're all getting caught up in the smaller blood vessels. They all like lock in with each other and create these clots. People with sickle cell disease have a very high mortality rate. Sickle cell disease is caused by a base substitution. We know what mutation causes it. So here's a normal hemoglobin gene, and here's the mutant hemoglobin gene. That huge problem with the shape of the blood cells in people with sickle cell anemia, that it all comes down to just this one base substitution where one amino acid was changed out for another. Gene expression is a really big thing. I know that was a big video with a lot of information, but now you know how a molecule like DNA can carry a genetic message and how that message can be manifested as a living organism. It's just a bunch of code for building protein. And proteins build cells. They connect cells together. They are communication molecules that allow those cells to communicate with each other and cooperate with each other. They're defense molecules that can protect those cells. They allow the cells to move. They're like little motors that allow the cells to move about. All those things that proteins do. So when you put all of that together, those proteins can build a living organism. And it's the proteins that are encoded in the DNA. The next and final video in this lecture series is on gene regulation.